Hey folks, welcome to the final lecture video of the PARE-Seq program. We're so excited that you've stuck with us and put up with even me talking to you for this many minutes. But here is where we get to apply what you've been learning in this short course, discovering what antibiotic resistance genes are present in the metagenomic sequence data from your soil or water sample. To do this, we'll use bioinformatics. Specifically, we'll use the Galaxy platform to analyze files generated from nanopore long-read sequencing, as we've discussed in the previous video. Then we'll talk through analyzing all the cool data the bioinformatics programs have compiled. In this video, I'm going to break down what the workflow in Galaxy does with a DNA sequence. It's a compilation of bioinformatics programs put in an order that provides the specific outputs you're interested in, namely what antimicrobial resistance genes, or ARGs, are present and which bacterial species they came from in your sample. We've created this custom workflow for you, so all you do is input your sequence data in the FASTA format. Then each program runs in the right order. Last, we'll talk through each of the 4 key final outputs: the summary statistics tool, the ARG alignment table, the taxonomy pie chart, and the ARG taxonomy table. You will display and analyze these visuals in your final projects. You're going to leave this video with a number of different bioinformatics analysis skills. We've listed them here all in one place, but throughout this video, we'll make sure to tie each concept back to one of these 5 learning objectives. By the end of this activity, students will be able to explain why the concepts of evolutionary conservation and homology underlie bioinformatics research. 2, assess the distribution of read lengths in a metagenomic sample. 3, interpret bioinformatics outputs to identify the antimicrobial resistance genes present in a metagenomic sample. 4th, to interpret bioinformatics outputs to identify the predominant species present in a metagenomic sample. And 5th, list the steps required in bioinformatics to identify an antibiotic resistance gene type from an environmental sample and match it to its host species. We're going to begin with a discussion of homology, a concept that underpins how Galaxy can identify a specific ARG sequence in a long FASTA file of DNA A's, T's, G's, and C's. Then we'll move on to the bioinformatics workflow and how it works, followed by an explanation of the 4 key outputs. After this video, you'll follow either the Galaxy video tutorial or the written Galaxy instruction manual found on the PARE-Seq or PARE website to enter your sample FASTA files and get analyzing. By the end of the Tools for Bioinformatics video, we had discussed that our data for PARE-Seq comes from nanopore long-read sequencing, and a FASTA file is the sequence of DNA bases present in your environmental sample. But how exactly does Galaxy sort through this sample to tell us what antibiotic resistance genes are present and which species of bacteria they came from? In bioinformatics, we use computer programs to sift through all the nucleotide sequences in a metagenome to find those that look like known ARGs. The programs align the sequences from our entire FASTA file to a database of ARG sequences to perform a homology search, looking for DNA sequence similarity. Alignments are scored based on the number of matching nucleotide bases, taking gaps and mismatches into consideration. A high score suggests evolutionary conservation. And in our case, we're searching for sequences in our FASTA file that show alignment with a gene sequence known to code for antibiotic resistance protein, antimicrobial resistance, or ARG sequences. In the case of PARE-Seq, the ARG database we will use was created for this activity to include sequences of the 5 resistance genes or gene families we discussed in video 4, Spotlight on Antibiotic Resistance Genes of Clinical Significance. When we see that the alignment tool in Galaxy has identified a specific sequence, then we can infer that this sequence is an ARG because of its close sequence similarity or alignment to a known ARG. In theory, you could search your metagenome against any database. However, because of the huge amount of sequences to sift through, running programs to carry out these types of searches can take several hours or days. Usually, they are run on supercomputing clusters. In order to run your sample on Galaxy, we are just assessing the presence of 5 resistance gene families, and the samples provided are each small portions of the total metagenome of the soil and water samples. Take this sequence of nucleotides, for example. Let's say that you know this is a known sequence of an ARG. Your Galaxy workflow will detect when a homologous match appears in the FASTA file you inputted and outputs that sequence as a potential ARG in your sample. Think of this process like a needle in a haystack, like we talked about last video. Likely very few of these sequences homologous to known ARGs exist in an entire metagenomic sample, but using bioinformatics, we can pick out these sequences and detect the presence of antimicrobial resistance. Now that we've discussed homology, the underlying concept of bioinformatics, let's go over what a Galaxy workflow actually is. Remember, each of the bioinformatics programs in the Galaxy workflow carries out a specific task using the sequence data that you've entered. Here you can see an overview of the PARE-Seq workflow on Galaxy. Think of this like a road that diverges in a number of different ways from an origin point. On the far left, we see an input box, which is where the soil or water sample FASTA file gets entered. From there, the file is run through Nanoplot, a summary statistics tool. It is also run through a taxonomy identifier tool, and then onto a tool that outputs a percent abundance chart of all of the species present in your sample. Thirdly, it is put through an alignment tool, as we just discussed with the concept of homology, which will align portions of the metagenome to ARG sequences in a database and output a table of these ARGs. Let's now go through the 4 key outputs you will analyze from this bioinformatics analysis and what tools are used to produce them. The Nanoplot or summary statistics tool will produce a series of outputs including a histogram of sequence read lengths and a table displaying mean and median read length, total number of bases, and total number of reads in your sample. This data helps us to assess the distribution of read lengths in a metagenomic sample, or learning objective 1. Notice here that hundreds of thousands of bases are sequenced using the long read method, and the average length of a read is over 5,000 base pairs, like we discussed in the last video. The median read length is a good assessment of the central tendency of read length for your sample. Means, as you may have learned in a statistics or other math class, can be skewed by very large or small values. Moving on to the alignment tool, this tool aligns the FASTA file you inputted to the resistant gene database we built. Here on the left, the yellow line represents the genome of just one bacterial cell in the entire metagenomic sample. The green chunk represents a tiny portion of that genome containing an ARG. On the output table, moving from left to right, you will see the ID number of each read, next the sequence length, followed by the nucleotide start and stop positions, and then the name of the antibiotic resistance gene identified. These are the outputs for the water sample, and you can see the ARGs that are present include a variety of resistance genes. Remember, there are over 50 kinds of identified tetracycline resistance genes. Taxonomy is the classification of the species represented by the DNA in your sample, i.e., what bacteria are present in the soil and water that we sampled. We can do this because different families, genera, or species have different characteristic sequences. The taxonomy identification tool uses a sophisticated alignment process. In this case, the tool scans the metagenomic sequence to identify the species of best fit using a complex algorithm. Comparison is made against a standard taxonomy database operated in Galaxy with all bacterial, fungal, archaeal, and eukaryote species they have on file. It outputs all the species it can find from our water and soil samples, and this is the table that it produces. Keep in mind, although I'm referring to species, these types of analyses are rarely able to predict at that level of detail. More than just DNA sequence information is usually necessary to assign beyond the level of genus. The first column, listing either C or U, tells us whether the taxonomy was classified or unclassified, i.e., whether the program was able to match the sequence to a known organism represented in the database. You can see circled in red the species ID. Some just say bacteria because it is as specific as the bioinformatics program could get, but others read Pseudomonas aeruginosa or Escherichia coli, all different types of bacteria identified by their genus and species names. This is a really interesting table just to explore. Read the Latin names of the species and look up any that seem of interest to learn more about their characteristics. The next graphic we will discuss is the taxonomy visualization tool. In this interactive pie chart, we will see what proportion of the total DNA sample comes from each of the different species present. This tells us what taxa are most abundant in our sample. On Galaxy, this is a very interactive tool, though you'll only use a screenshot likely in any final project you complete for this short course. On the Galaxy website, you have the ability to explore the percent abundances of each kingdom, phylum, class, order, family, genus, and species in your samples as you click through this chart. By clicking on the largest portion of the pie chart repeatedly, this might take some trial and error. Look at the upper right-hand corner of the screen to see what percent of the root each portion you click on is, and choose the largest percentage each time. You can get down to the genus level and find the type of organism in highest abundance in the sample. Essentially, this visualization tool takes the data from the taxonomy identifier tool and displays the proportion of reads with a sequence aligning to each particular taxa. A few things to note when you explore this chart on Galaxy. Getting down to the species level will still only be a portion of a percent because there are so many different species in this entire sample. And also, you can see the small proportion of the metagenome that viruses or eukaryotes, like nematodes, fungi, protozoa, and us, humans, make up. Viruses on this chart are purple and eukaryotes are green. So now you're at the final part. Though not as visually appealing as the pie chart, this table really is the culmination of this analysis. We will determine which bacterial species are harboring the different antibiotic resistance genes that are detected using the ARG Taxonomy Table. At this point, the workflow will merge the alignment tool homology search for antibiotic resistance genes with the taxonomy homology search results to form an ARG taxonomy table that will show you the host origin species for the antibiotic resistance genes present in your metagenomic sample. We first scanned the entire FASTA file for regions that align to ARGs in our reference database. Then the FASTA file was scanned using a sophisticated algorithm that requires a ton of computing power to look for various sequences that are characteristic of different taxonomic groups. The next bioinformatics program merges the data from these earlier steps to link the taxonomic information with the ARG information to create this table. You can see, for example, TET-G in this sample comes from the same read predicted to be Pseudomonas aeruginosa bacteria, or maybe in a plasmid that came from these species, for example. This gives you a picture of what bacteria harbors what resistance genes in your sample and allows continued environmental surveillance for these potential public health threats. So it's easy to get lost in the weeds when taking a deep dive into an area such as bioinformatics and antimicrobial resistance. But let's take a minute to put your findings from conducting this experiment in Galaxy into perspective. This type of environmental surveillance work, identifying what types of resistance genes are present in a sample and knowing what bacteria harbor those genes, can help us to understand exposure routes. For example, when people are exposed to antimicrobial resistant pathogens, is that occurring through food they ingest, contact with soil, or in some countries through drinking water? Metagenomics and bioinformatics can also help us monitor the presence and abundance of specific types of antibiotic resistance, which provides information on emerging trends and potential human exposure risk. We can then develop interventions that prevent this exposure and increase antibiotic-resistant infections. Before setting you off to enter your data in Galaxy and perform your bioinformatics analysis, let's wrap up with a summary of what this video covered. We began by discussing homology and how this concept is involved in aligning our soil or water metagenome to an antibiotic resistance gene database to determine what ARGs are present in the sample. Next, we provided an overview of a bioinformatics workflow and how we developed a PARE-Seq specific workflow that will take your input FASTA file and produce a number of output figures and tables that help us analyze the ARGs present and from which bacteria they came. We dove into greater depth on specific outputs from the workflow, a summary statistics tool, the taxonomy tool, the alignment tool, the taxonomy visualization tool, and the final ARG taxonomy table. We explained what the outputs of each of these tools were and how you can gain valuable information about your soil and water samples from each. Finally, we put what you're about to do using Galaxy into context. It's easy to get lost in the weeds when you get this far into an environmental surveillance project, but let's remember that this whole process of detecting antibiotic resistance genes in a sample is like finding a needle in a haystack. And in the greater world of public health, it helps professionals determine where AMR has spread and how to curb its impact. So you're finally ready. As mentioned at the start of the video, head on over to the Galaxy Video Tutorial or Galaxy Instruction Manual on the PARE-Seq website, and you can get your account up and running to analyze your data. After finishing the bioinformatics activity, please fill out the post-course evaluation survey, found also on the PARE-Seq website. Until next time.